Thank you, Bailey. I'm sorry about this. Okay. I'm so sorry about this. Thank you so much for starting it. I think I didn't get onto the Zoom extension account right.
Sorry, I missed most of that.
No, you're good. Sorry. Just thank you for starting the meeting.
Oh,
I
yeah.
realized my account privileges were a little weird.
I can't figure it out.
Jesse, hi, and welcome to
Hello.
UVM.
Hi, how y'all doing?
Doing well. The sun is
Good.
shining over here.
That's always good.
Yeah.
After a long winter.
Yeah. Hopefully it's not a false spring, but we'll see.
Yeah, no kidding. How are you?
Doing all right. It's been a hectic week, but yesterday evening I got a nice email from the campus where it was, oh, internal grants have been pushed back 2 weeks. So more time for that.
That's great. Yeah, we just got an NSF like extension notification for some stuff that we couldn't do last year, and I'm like, oh, thank goodness. I think there's lots of grace around work right now, hopefully.
Yeah. That should be that.
Oh, nice. We've got interaction.
Oh yeah.
Awesome.
I was gonna have like little quizzes and like think pair shares throughout it since I'm at a teaching college now, but I was like, no, it's too much work.
That's fair. Yeah, I know someone who's in a similar position right now, and I imagine you're just swamped with, is it all virtual or some in person?
This semester we're half and half.
Okay.
Or well, we have to have at least half of each department's classes in person. So the course I'm teaching, which is upper div introduction to just like general biogeochemical theory, that's in person for the lab, online for the lecture.
Okay.
Are you all online or?
Yeah, we're kind of mixed up as well. I'm teaching a course, getting 2 semesters of TH about through bargaining, but it's all in person, and yeah, but it's a small class. It's only 9 people, so that's been all right.
So you're able to like socially distance and everyone's wearing masks.
Yeah, they've been really good about it, which is cool. The only thing is like lab is always a little hard to, I mean, since it's all indoors, everyone's kind of doing a COVID dance, I feel like.
Yeah. Well, I mean, I think so far a quarter of my labs have been outside to help with that.
Wow. That's amazing.
Told them you have to bring warm clothes on the days we say we're going to be outside, because we're going to be outside.
I keep trying to like come up with excuses. This is soil chemistry, so it would have to be a large excuse to get us outside, but I keep wanting to like do something.
But it's like the perfect excuse, like go outside and we'll dig some holes. And because it's still frozen, it'll take forever.
Yeah, the S horizon.
So your PhD, so you moved on to Candidacy, right?
Yeah, I did my comps at the beginning of the pandemic, which was fun. So.
Although it almost feels like that's better than having to do them like now or during the pandemic.
Yeah.
Anyway, congrats on passing your comps.
Thank you. Yeah, it feels good to have this behind me, although now I feel this like pressure to start writing stuff, and I feel like I'm still in the middle of all the data gathering.
Yeah. So do you do your qualing after at the end of your second year or third year? When?
It depends a lot around here. A colleague, she just did her comps, and she's already published most of her dissertation. But I I did mine at the end of my second year. So yeah, end of my second year.
So you should be in the golden year, right?
The golden year feels like the stressful year.
Well, yeah, because it's a pandemic.
Yeah, that's true.
Hello, third person.
Oh yeah, sorry. This
I
is
don't see
Bailey.
names anymore.
Bailey, this is Dr. Bateman. Dr. Bateman, is Jesse okay as well?
Yeah.
Okay.
Preference with Jesse.
Okay, good to know. Cool.
Nice to meet you, Jesse.
Nice to meet you as well. Why are you not giving me names anymore? Oh, well.
Oh, thanks for making me co-host, Bailey.
Yeah, the both of you should be co-hosts now. So
Great. Oh, and that means I can admit people too. So I don't want to. If you have stuff to do, Bailey, I'm happy to manage that. And
sure thing. I can do it while you introduce Jesse, and then
that's great.
if you want to take over.
And
from there.
usually we start like a couple minutes after, right? I thought I remembered that.
That's what we've been doing at like 1:05. Just because I don't know if people have been taking a long time to trickle in.
Yeah.
So,
Well, time is meaningless.
but yeah, I mean, pretty much.
Pandemic in time.
Perfect for chrono sequences, right?
No, the pandemic is perfect for nothing.
You mentioned like a kid, right? Are they back to school in terms of in person?
No, so our child is almost 2. They'll be 2 on Earth Day. So
Oh, what a great birthday.
yeah, worked out pretty well. Yeah, so they're at home, pretty much just running around in the snowbanks, and they keep they've been helping us with the maple tapping, so they like to hold the sieve to sieve out the ice. And so like half the time they're just like walking over to where the sieve is hanging, just pointing at it. Let's go outside. We have to check the trees.
That's awesome. I love that. My uncle just mailed me some maple syrup from Missouri, and I'm excited to. I'm gonna test it. Do like an experiment with Missouri and Vermont maple syrup. Although Vermont people here are probably, yeah, gonna make fun of me for saying that.
I support it.
Thank you, Bailey.
Wherever you're from, or wherever someone has sent you maple syrup from.
Yeah, it's got the like terroir of home.
Yeah. That's it. Oh, and for folks trickling in, if you would like to respond to this poll everywhere, please feel free to. I think the where to respond is showing, right? I can't actually tell what folks do.
Yeah, we can see the URL to respond and then also the text.
Okay.
So yeah, for folks, I can actually just throw the link to the website in the chat.
I like word clouds. They're so cool.
Definitely. All right, I'll give it another 2 minutes before I introduce you. Welcome, everyone. And there's a link in the chat if you want to contribute to Jesse's question, why do you care about soil?
I like thinking that the because is not because it was used in a bunch of different ones, but a lot of people are just like, because the soil is so universal, just because.
Exactly. Oh, yes, I can share it. Okay, welcome everyone. We'll go ahead and get started. I'm really excited to introduce today's speaker of our seminar series, Jesse Bloom Bateman. So Jesse grew up in rural New York with a great deep love for the natural world and a curiosity about the processes that shape it. For college, they traveled to New England to pursue a degree in geology and biology at Brown. There they worked in a terrestrial biogeochemistry lab, exploring plant-soil interactions, particularly on seedling uptake of phosphorus. In their doctoral work at Stanford, they investigated climatic controls on soil development and nutrient cycling. And as a postdoc, they worked on developing isotopic methods using pedogenic and mollusk-derived carbonates to understand paleoclimates. Jesse uses their background in terrestrial ecosystems to investigate climatic controls on biological communities. So for that work, they use a combination of isotopes, statistical analyses, and geomorphological studies. So currently, Jesse has returned to Central New York, where they are a diversity faculty fellow and assistant professor at SUNY Cortland. In this, they are continuing their previous work and also supporting undergraduate students' exploration of the scientific process. In addition to their scientific pursuits, Jesse works to create a more inclusive and diverse STEM ecosystem. In this arena, their work includes outreach in the broader Los Angeles and Central New York communities and inclusive pedagogy research in the classroom. So welcome, Jesse. Great to have you here, and take it away.
Great. Thank you very much for that introduction. Vic, happy to hear another SUNY affiliate out there. Great to see everyone getting out and doing great things. So as Jenny said, my name is Jesse. My pronouns are they, them, he, him. And today I'm going to be talking about some of the work that I've been doing from my PhD work and then into some of the stuff I'm still doing and future projects that I'm currently really hoping that people who think about soil and ecosystems would be happy to start some conversations with me about. Let's see, there we go. So as you were all waiting, you were kind enough to get us started on thinking about soil and why it's really important. This is the word cloud that was coming in off of your answers. I'm seeing a lot of things like carbon, sequestering as well, so carbon sequestration, I'm guessing, life sustaining, right? It's providing ecosystem services. So I feel like we've all in the same headspace, right? That soil is amazing and a wonderful portion of the ecosystem that we should all be thinking about and enjoying. And yeah, so like soil is, right, from a very selfish standpoint, we can think of it, soil as this medium for plants to grow in. It's the physical media in which roots are anchoring, as well as the source of most of the plant important nutrients that plants need to grow, and then we eat plants. So, right, we can be very selfishly thinking of those things that soil is giving to us. And then also, oh, okay, good, got clicked to go. Right, it's very important in carbon cycling and regulation of the climate, especially on sub-1,000-year timescales. So if we think about different pools of carbon where carbon is really actively cycling, so soil, vegetation, atmosphere, and upper ocean, right? Soil is the largest pool of that carbon, and how we treat soil and how we manage it / how it exists in natural systems plays a very strong role in climate feedbacks, as well as making sure we have a sustainable system. So the society being able to be sustained as we currently understand it. Now, those are like more like the high-minded, big social reasons to care about soil. I also have some very personal reasons. So as Jenny said, I grew up in central New York. The soils here aren't the best. They're mainly channery silts, 3 to 10 inches to bedrock that I grew up on. But I grew up interacting a lot with dairy farmers. Most of my best friends were on farms. And then I worked for the Natural Resource Conservation Service through my undergraduate. And so I really was developing a lot of this connection to the natural world, to the outdoors, and wanting to understand why landscapes look this way, why soil is the way it is. And so that has been this drive of like, oh, I get to go outside as a job and understand the natural world. Cool, sign me up, I'm there. And then in doing so, I've been able to interact with a lot of great folks. Come on. Is there? Oh, sorry. I'm not the best at doing this. Laser point. There we go. Some great folks. So this is us, the UCLA lab group, out collecting carbonates from paleo lakesides, trying to reconstruct high-standing lakes from the last glacial maximum in the Western United States. Right? And so like being able to do that field work together outside, digging soil pits, collecting carbonates off of rocks, has a very community building feel to it. And I've really felt a lot that soil science and ecosystem ecology has been great at developing community and not feeling like I'm this singular scientist shut off in one lab space all by myself. Although I do say that now sitting in a office all by myself because of a pandemic, which has been very rough. So when I'm thinking about soils, I'm really thinking about how soils are developing and how we can understand them. So, right, that's the soil forming processes. So the additions and losses that happen as we're going from pretty much bare rock or sediment into developing these beautiful, differently colored, differently textured, complex ecosystems. And as Jenny pointed out back in the 40s, like there was this idea that if we understood these 5 soil forming factors, topography, climate, time, biota, and parent material, if we could put together a function that could describe all soils if we understood those inputs, those controlling factors, right? And I'm not so bold or so long in the tooth to believe that I can look at all 5 of those. I'm mainly in my work focusing on 3 of those, so climate, time, and biology. And for this talk, we're mainly going to be focusing on climate and time, with biota sprinkled in as a flavor bit. So for climate, we can think about like external controls on climate, or external controls. I'm really thinking about it as water, like a controlling on water. And so we have water inputs. That water is controlling chemical reactions, right? It's bringing reactants to mineral surfaces to react with the physical structure there. It's removing chemical reactants. It's really the soup in which everything is occurring. Where it's also physically moving the material throughout the system up and down, so through translocation. And also, if you have gases, you have movement out of the system that way, or also you have gravitational losses through leaching, or losses through gravitational leaching. And so in a system that doesn't have any any vegetation with it, we might expect to see that nutrients through time will eventually equilibrate to moving downward in the system as water is pulling nutrients out of or it's weathering and reacting with minerals at the surface and drawing them down to depth. Eventually, if we let this proceed long enough, this would end up being a line that was actually just over here, as all of the nutrients such as phosphorus or calcium have been leached out of the system. Now, biology, on the other hand, is really adding a lot to, especially in ecosystems where you're not harvesting material out of it, right? It's adding organic matter, which is very important for the retention of cations and anions through exchange and absorption onto surfaces. The exudates from their roots, and yes, I know microorganisms do a whole lot on chemical weathering, but I'm more of a plant-focused individual. So root exudates and are helping with chemical weathering as they're releasing acids to break down the minerals so that they can access elements that help them grow. And then also the roots growing through the material is helping provide channels for water to move. Its roots are physically entering into rock and then breaking them open so that water can get in and reactions can occur. So they're also providing a physical weathering of the material that's there. And right, they work together. Climate and biota are going to also have interactions. It was just talking about them in isolation, but they also interact to work against each other in some ways and in conjunction with each other as well. So the water, like plants are able to manage or to force water into or out of the soil through deep rooting and pulling water to the surface. When they're doing that, oh, I see. Come on. Oh, that's weird. And when they're pulling water up through the system, up through the soil, and they're pulling in nutrients that they need from the soil, this is what we would call biological uplift. So they're bringing nutrients to the surface, it's existing in the biomass, and then when it falls at the surface, enriching the surface layers with plant important nutrients. So with vegetation, we end up seeing this sort of pattern where you have a enrichment of the surface layers and then a depletion at depth of plant important nutrients, right? And this is that interplay between the water inputs and leaching losses versus the uplift, the biology trying to cycle and keep their important nutrients there in the soil. Time, where climate and biology, I think of them more as like active active controls. Time is more of the integrator of other actions, right? It's the integrator of climate on the system. So as time proceeds, you have an accumulation of organic matter, right? If you have just newly erupted lava, there's little to no organic matter there unless it entrained some trees or other organic matter as it was flowing. But as you have colonization by the ecosystem, you have deposition of leaf litter, you have roots that are dying and then are accumulating in the upper layers as organic matter. You have a deepening of the weather profile. Those roots are driving down and they're expanding the chemical weathering front. And you'll also end up eventually having losses of plant nutrients. And this is where you end up because you end up having the leaching. Like, no system is a perfect system in holding water there. And water is always going to take some amount of ions with it. And so over time, you slowly lose nutrients from the system. But with the plant important nutrients, you maintain for a very long period this enrichment of the surface layer. And so that difference between the surface and deep soil becomes more pronounced in concentration of plant important nutrients. So with this sort of in mind, where are we going today? So first, I'm going to talk about some work that I had done identifying climate-driven patterns in soil properties and creating a conceptual model for how that worked based off quantitative analyses. Then I'm going to compare those patterns across soils derived from differently aged parent material. After that, there's some caveats to how we're doing that. And so a modeling project that we did to try and get at some of the differences we were seeing and whether or not these gradients are really water balanced, so water-based, or if it's more of a temperature-water combination. And then lastly, hopefully we'll have time for it, is getting to what I'm working on now and trying to better understand the effects of forest fire on carbonates that are stored in the soil column, especially how forest fires may impact the clumped isotopes of the carbonates. So thinking about soil, I'm a scion off the Vatusic-Matson tree. And so whenever we want to work on ecosystems, especially soil systems, I think of going to Hawaii, specifically the Big Island of Hawaii. It's a little selfish in that, you know, you get to work in paradise for 4 to 6 months out of the year, but it also has a lot of very important features that make it a well-constrained system for asking questions about soil development, because you can either keep the soil forming factors constant, or you can vary them in a fairly systematic way. So I did most of my PhD work looking at Mauna Kea volcano between about 4,000 feet and 11,000 feet of elevation. There's a lot of hiking up and down, which, so much fun. Such a beautiful place to go hiking. And right, so for the soil forming factors, ones that I wasn't directly looking at, parent material and topography, parent material is very consistent across the Hawaiian archipelago because it's derived from hotspot volcanism. And so Mauna Kea volcano is a basaltic core that has an overlaying of volcanic ash largely over it. And that's most of the Hawaiian chain of volcanoes, is basaltic magmas with volcanic ash. And geochemically, they're very uniform across the volcanoes, with 2 tracks that have slightly different geochemistry of their magmas. Topographically, right? I'm saying, okay, we can hold this fairly consistent, but it's a mountain. So what we're doing in sampling on Mauna Kea is looking for topographic high points, so local highs. And what that does is it minimizes erosional inputs and depositional losses. So we're not really describing accurately the soils of Mauna Kea. It's much more than describing a volcanic soil whose topography is close to flat as possible. So moving it out of the local space and into more of a theoretical space. Yeah. For the factors that I am interested in, for climate, it is topographically driven and has been fairly stable through time, especially over the past 20,000 years. It ranges from about 250 millimeters of rainfall up to about 4 meters of rainfall on the gradient. Yeah, so that ranges from drier to Stockton, about drier than Stockton, California, to 5 times as much rainfall as Seattle to get a more continental perspective. And then for time, Mauna Kea is roughly 20,000 years since the end of ash deposition. So that's what I've used as this is a valid time point of, we're going to call this 20,000 years old. Yes, soils were developing prior to that, but that is the end time of really like pretty much the end of actual depositions that were widespread on Mauna Kea. And later this will become important, is that we can go from 20,000 to 4.1 million years by looking across the island chain. So this is the chrono sequence. On Mauna Kea, I have a climate sequence on the 20,000-year-old, and then looking across the island, we end up with the chrono sequence. And then even though like across 7 islands, the biology of Hawaii, because it's so remote, is fairly low in biodiversity. Thinking about a handful of major canopy species, and so plant biodiversity and animal biodiversity. I know someone recently looked into the microbial biodiversity across the gradients, but I don't have that off the top of my head, unfortunately. But yeah, microbes are probably going to guess are actually probably fairly diverse across everything. But yeah, we go in the very dry sites on Mauna Kea to no plant life, no vascular plants, into a savanna mamane, which is a small wooded brush type tree, into koa forest, a large nitrogen fixer, and then eventually into ohia dominated forest. All right, so starting with that first idea is identifying climate-driven patterns in soil properties. To do this, right, you got to go out and dig a whole lot of holes, collect a whole lot of samples. And there's 2 types of samples that were used. The first type I used were 30 centimeter cores, where dig down 30 centimeters and then integrate the entire core for the sample, taking equal volume from each, not equal mass from each. Across approximately 14 kilometers of transect, I took 97 cores spaced at 250 meter intervals for the central portion of the transect, which you can see right here, where we thought where most of the change might be happening. That is 2 transects that run in parallel, but then the tails out into the very dry and the very wet were singular transects. That was partially because, right, we thought that most of the change we might be seeing is in the central portion of the range, and then also just a feasibility of sample analyses, of collection. And then on top of those 30 centimeters, I also took 8 soil pits, dug 8 soil pits where it did depth sampling by horizon. Those data didn't really, they informed understanding of what the soils would be categorized or classified as at each different region, but this went much more into a study on the solid state of the soils there and the iron and aluminum dynamics, which currently still working on revisions of that manuscript and not talking about that today. So got our soil collected, back in the lab, pretty much a fairly basic suite of analyses, really looking at the bulk chemistry of the soil, carbon and nitrogen content, base saturation, so how much of the exchange sites are filled with base cations, cation exchange capacity, so the amount of exchange sites per mass in the soil, pH, plant available phosphorus. So for that, it was just a resin exchange extractable phosphorus, and then exchangeable cations. So again, resin exchange cations. Now, I'm trying to identify what is the conceptual model that I'm using when thinking about identifying climate-driven patterns. So in the early 2000s, one of my mentors, Oliver Chadwick, and another co-author, John Chorover, were thinking about how soils like transition, and they came up with this way of thinking of, or how I was exposed to it from them, pedogenic thresholds and soil process domains. So thresholds are, and this comes from the idea that soil is like a buffering, multi-level buffering system. And so thresholds are where one of the buffering portions or a portion of the system that keeps it in a balance is either runs out or comes into abundance. And so you have a rapid change, that's nonlinear shift over a small change in external forcing. So small change in, say, rainfall or age, and you have a rapid change across that. On the flip side of that are soil process domains. And these are regions where soil properties or processes are fairly constant. And so you don't have as much change within, if you're looking across a range of the environmental variable, you don't have as much change in either property or process. And so take a second what that looks like. You have, right, so process domains are going to be these 3 colors, the light gray, the blue, and the darker gray. And then the thresholds are the boundaries between the domains. Here, so this is a figure that I've redrawn with the process domains overlaid on it from Kohala. So this is the 150,000-year-old climate gradient. The thresholds that they were able to detect are uplift. So you're first seeing biological uplift in the soils. And then at the wetter end, you're seeing the depletion of primary minerals in the surface soils. And then the 3 process domains that are then bounded by those are a wind erosion dry process domain where you have development of carbonates, biological uplift dominated and mineral weathering dominated central domains. So this would be high plant fertility for the soils. And then eventually iron enrichment, and you have low plant nutrients because they've all been leached. And when we plot the data over those, so this is plant available phosphorus, we see that it's low in this wind erosion dominated process domain. We have a sudden increase in the amount of it into the biological uplift domain, and then you have depletion again. And so this is what we're looking at, is the balance between climate and biology. In this central domain, you have biology being able to enrich the surface soils relative to the amount of leaching you have occurring due to rainfall per site. it's important to think of when looking, each one of these dots is a different site. And if we were to think about this, like how do these soils evolve over time? It, while it took me a while to get my head around this, but like, I want to be like, oh, as soils move from dry to wet, but it's actually, instead of thinking of these being able to move left or right, uh, sites being able to move left or right, it's really that they're moving into and out of the screen, and that's because the z variable here would be time, and that all of these sites have their own trajectory, and a young site that is in a dry and then moves to a wetter is going to look different based on its trajectory than a site that was young and fairly wet and then moves to stay that same wetness. Yeah. Which we've been trying to do some modeling and trying to understand, like, can we predict soil collapses? And that hang up makes it a little bit harder. But it's been an interesting conversation to be having with folks. All right, so about to get into the data. Before we do, as you saw in the last slide, the variable of interest was rainfall. And so rainfall would be a water input. I've really been pushing, and I've convinced at least my little research impact space that it's probably a better way to think about it is actually water balance. So water inputs minus losses through evapotranspiration. And the idea here being that if we're thinking about like gravitational leaching or what water is available for chemical reactions to be occurring in, you're not actually caring just about what water has come in, but what water is potentially just there. So the water minus the gaseous losses is much more, in my opinion, representative of that idea of what's actually interacting with the system. Also, if anyone here is a hydrology or a hydrologist or taking a hydrology course, this is not the hydrology water balance. It's more of like a simplified hydrology water balance in that it doesn't take storage into account or runoff into account. So while I'm making this argument that, oh, we should use this because it's so much better, we'll also say that on apologies, on Mauna Kea, the difference is really just an offsetting because potential evapotranspiration is fairly constant across the volcano. Precipitation, which is the dark blue, versus water balance, almost the exact same pattern. You just have an offset because of the water balance. So especially because most of this work was done when I was a PhD student or had just recently graduated, it was kind of like, okay, how can I convince people of this patterning that late stage full faculty had said is present? And I'm younger, I'm not going to have the same clout, so I can't just pull a Walker and Cyrus and put up a figure that has no labeled axes. I needed to come up with a quantitative analysis for it. And so a lot of my work was applying this novel technique of using segmented regression analyses to the data to see if I could use those to identify qualitative thresholds and process domains. And so this is the data again from Kohala using water balance now instead of just rainfall. And with segmented regression, what we're doing is it's a broken stick regression, linear regression. It's naive in that we assume the linear with no breaks is the best fit. And then, excuse me, if that's not true, I would break it up and go, okay, does one do it better than 2, or does one break do it better than 3? Looking to see which one had the best fit if a unbroken regression did not provide the best fit. It allows then to figure out points where you have those breaks, and then also confidence intervals for them. And so if these confidence intervals overlap, we could argue that, oh, that's unlikely to actually be 2 thresholds because we can't actually pull them apart in the data, which ends up being a bit of a limitation of the of this method, but it also prevents us from overfitting and trying to say, oh, there's all these different thresholds and peaks that we can find. So looking at actual data, so these are data from Mauna Kea. On the y-axis, we have silicon, aluminum, and iron. And then on the x-axis, we have water balance. What this 0 means is that that is the breakpoint of inputs minus outputs. So that is where precipitation balances evapotranspiration. And so for silicon, we see that there is a threshold around 1,000 millimeters per year of water balance. For aluminum, there is no, it's just a consistent decrease. And then for iron, right, there's no losses or gains on average, pretty much. It's just an overall pretty even, no change. The reason it's slightly elevated over 100% remaining is that you have a slight collapse after the loss of others means that you have a slight over retention of iron. It's not that iron's being added to it, it's that other materials have been removed from the system. Using calcium as a representative of the base cations, all the other base cations, or the base cations largely follow the same patterns. And then, right, in percent remaining relative to parent material, about 740 millimeters per year. And then the confidence interval also overlaps 1,000. That's also the same for concentration in soils, in surface soils. But when we look at exchangeable, what we see is that there is a peak, and then there is a another break higher up. And so we have 2 breaks in the calcium's exchangeable, as well as in magnesium that shows up as well. Base saturation and CEC. So CEC is increasing. This largely increases with organic, or with the carbon increases, but then has a decrease as we get wetter and wetter. And then for base saturation, it follows the pattern of calcium because the dominant cation for Mauna Kea soils is calcium. These soils are all acidic, and they just become more acidic as you go wetter and wetter. But we do end up getting a threshold that's about 940, but it has a fairly wide confidence interval. And so using these data, right, I'm trying to be careful, and I might slip up from time to time, but I'm not saying that these are thresholds in and of themselves, and that if one property changes at that point, this just allows a quantitative assessment of, okay, do we have a lot of properties changing at the same water balance? If so, what does that mean, right? So we have to pair that quantitative analysis with a conceptual understanding of the system. And so that conceptual understanding that it developed, right, is that about 750, if you average across all the different properties changing, you end up getting a threshold that would represent the depletion of primary minerals, right? So that's around the point where you have the change point in pH. That is the point where you have depletion of calcium and magnesium, right? So this is the water balance where over 20,000 years of soil development, water is able to leach away those elements such that you're no longer having an enrichment of the soil surface above and when you're in wetter spots. When you're in drier, oops, did this backwards. Yeah, so when you're drier than that, you have biological uplift and mineral weathering. So you have high fertility, right? They're balancing each other out. Enough water is present to provide the weathering of the minerals and the plant nutrients, but not so much that it's all being removed. And then, as I said already, right, above, when you're wetter than that, that's when you end up having supply limitation. But iron not being as readily or as mobile in the system becomes overall enriched there. So you end up having soils that are very red, right? Typical oxasols and altisols. And once you weather everything else out, you're sticking around with a lot of iron and some aluminum present. So that's what I was able to identify quantitatively. And then I spent the last year and a half trying to convince Peter, my dissertation advisor, that there was something else there. This little ball of sites that I found very confusing is that seems like something's there. And so what is plotted here is the percentage of calcium in the exchangeable pool relative to the total calcium. So this is exchangeable over total. And to me, what this was trying to get at is how much is actually getting out of minerals onto surfaces versus how much of it is being bound up in primary minerals, secondary minerals, or organic matter. And what we see is it's very low and then it shoots up, and then until you get out into the low fertility, it stays fairly high. But that being said, even though we have low exchangeable, we still have a lot of calcium there. It's not just that, oh, we're losing all of the calcium that's present once it weathers out of a mineral or once it's broken out of organic matter that's occluded in, which would be what I'd be more likely to interpret out here, right? Like it's just been pretty much lost from the system. No, this is that it's there, it's just not out into plant accessible forms. It's still in physically bound up and stuff. And so arguing that with him and saying look, I can't identify it quantitatively, but there's something there at about minus a meter of water balance. And I think what that is where I'm picking up the threshold in mineral weathering, right? Where there's enough water that's present, there are vascular plants that are present that are able to drive the weathering of the minerals at a rate that's high enough that you actually see that signature in the soil, right? So below that, you have little weathering of minerals, and then anything that does come out, because you don't have a lot of biology present, any pulses of water have the potential to just drain it all away, right? And so that's more, instead of it being supply limited, this is the kinetic limitation on fertility, right? Because there's so little weathering there, you don't have much of an input of plant important rock-derived nutrients. So this high fertility is bounded by low fertility, but it's two very different systems that are dominating that low fertility. On one end, it's kinetic, which if you end up having more water coming in or there's more biology, you can reverse that low fertility. But on the supply limitation, unless you have a new input of nutrients, that's what's there. It's an irreversible low fertility. All right. So from that, right, able to demonstrate the feasibility of the statistical method, yay! Was able to get my PhD, yay! And was able to identify one threshold statistically, but then also identifying the limitations of this, and that once we get close to the edge of the range, the boundary conditions of trying to identify a threshold become very difficult. All right, so got this pattern, got this conceptual framework, quantitative analysis that works. I'm going to apply it across the chronosequence. So got first step, I controlled for time. Now I'm going to vary time. So from down to just over 1,000-year-old soil, or soils derived from just over 1,000-year-old parent material, up to soils that are derived from 4.1 million-year-old parent material. And we have differing sizes of rainfall or water balance gradients, from fairly small to, Mauna Kea. I kind of put in the work. It's the biggest one. Not going to brag, but going to humble brag. So we've got this idea. This is Mauna Kea's different patterns, or this is the Mauna Kea conceptual model. Now we're going into, on the y-axis, parent material age. So we're going youngest to oldest, but then still on the x-axis, we still have this climate sequence. So now we're in an age climate matrix. And what I found was the first early stages, their gradients are too short to really perform the quantitative analysis. The soils are somewhat enriched in plant important nutrients. However, because it's so short, like we don't actually capture either side of thresholds. So you can't say whether or not this is a process domain or if it's two and they just haven't developed yet. But if I had to put them in one, it's like starting to see biological uplift. Then on Mauna Kea, which is 20,000 years old, we have quantitatively identified biological uplift and iron enrichment. We see this as well in the 150,000-year-old and 4.1 million-year-old sites. So also in the 4.1 million-year-old is able to quantitatively identify the iron mobilization threshold that Oliver had identified in his work. And then in the 150,000-year-old was able to identify the wind erosion threshold. So really cool thing that happened with doing this is that the quantitative analyses and the qualitative analyses that had previously been done by Oliver and Peter agreed fairly closely with each other. And that what we see is that where we can identify this biological uplift pattern, we actually see an overall decrease in the water balance necessary to hit that threshold. So this is looking at, think about like, okay, the integrated amount of water passing through the system is able to leach nutrients out. And the longer you're having that like little bit of leaching, you're able to end up having it be in drier and drier sites that it shows up. So you have low fertility getting drier and drier sites. Yeah, so, sorry. Primary mineral exhaustion threshold negatively correlated with parent material age. 20,000 year gradient contains the transition from kinetic limitation to supply limitation, right? So in one gradient, we're actually able to go from this kinetic limitation, no limitation on weathering that we could find, to a supply limitation, right? It was really cool to have, and also provides this insight like, oh, what other interesting questions can we ask? And then happily the methods agreed with each other. So little plug shamelessly, this is the paper that published that in. Please read it, cite it. If you want to have conversations with me about it, happy to talk about it. That being said, the last page is one big old caveat. So this is a map of rainfall on the Big Island of Hawaii. Red is dry, blue is wet. That's all you have to think about for it. From that, Kohala, which is the 150,000-year-old site, versus Mauna Kea, the 20,000-year-old site, very different patterns. Look, we can see, so Kohala is on the windward or the leeward side, so it is a rainfall shadow desert formation, whereas Mauna Kea is on the windward side, so it's the stacking up orographic uplift driving the rainfall. That also means that temperatures are flipped. Kohala is fairly low change in temperature, but Mauna Kea is very cold where it's dry. So Kohala goes from hot and dry to slightly cooler and very wet. Mauna Kea goes from very cold, very dry in Hawaiian terms, to very warm, very wet. I'm wondering, what is the balance between control of temperature versus water balance? And is there modeling we could do to try and get at that? There we go. So I'm gonna finish up, with this. Sorry, I just saw the chat from Jenny. Apologies. So pretty much set up a model, looking at the different components, breaking it down, controllers on temperature. The whole model itself is driven by rainfall inputs, and then we can turn temperature on and off to control plant productivity, weathering, and nutrient availability. For Mauna Kea, we're running at 60,000 steps, so monthly time steps that's about 12,000 years if we think about it like that. So it's a shorter run, and then we run it for 10 times as long to see if time is the driver and we can just set those up without temperature, or if we end up getting different patterns when we include or do not include temperature. For Kohala, so the 150,000-year-old, what we're able to do is looking at calcium and phosphorus. Solid line is phosphorus modeled, dotted line is calcium modeled. Actually does pretty well. Okay, so we're able to pick this up. This is when we do or do not use temperature in the system. So what happens if we turn temperature on, which we did it as kind of backdoor envelope, back of the envelope as just a scalar. So it modifies it based linearly off of water balance because they're so correlated. And what we find is that, there's change, but the overall pattern is still the same. If you include temperature, you get a bigger increase enrichment in phosphorus, and you get a slightly later decrease in the calcium. But when we look at Mauna Kea, the pattern changes entirely, especially for phosphorus. For calcium, it doesn't as much. And so it's not just that this is a magnitudinal change, it's that the pattern itself actually changes. And so what we're getting at is that on the 150,000-year-old, we can pretty well understand it just through water balance. But because Mauna Kea is higher elevation and the temperature change might be that it's just at that boundary and where temperature becomes important, we're unable to say whether it's a temperature or water balance, right? And we can't conclusively say which one is the stronger control. All right. And I believe my time is done, so I'm not even going to tease you with those and just be like, so where did we go? We talked about the quantitative identification of climate-driven patterns, established the conceptual model, and then looked across a climate age matrix. And then we really, for the temperature versus water balance control on Mauna Kea, the jury is still out and trying to understand if it's a pure water balance gradient, which it looks like we're not able to say that. The temperature is going to play a pretty important role, but what that role is, we haven't really dug into that. All right. And with that, I would like to thank my co-authors, folks that I've been able to bounce ideas off of in both the Vitusic lab and the Tripodi lab. Didn't actually get to talk about any of that stuff. Sorry, didn't time it well. And then also the folks who have helped me in the field and all the very helpful and generous support financially that I've received over my career. For that, questions?
Awesome. Thanks, Jesse. That was great. I love the last photo too. I have a picture like that of my own.
You have to have a photo of yourself in a soil pit.
Exactly. Awesome. So for questions, feel free to leave them in the chat or unmute yourself and speak them aloud or raise your hand. I guess I'll kick it off. I don't have a ton of experience with some of these chronosequences and climosequences of my own, but I find it really interesting. And I know recently there's been kind of a resurgent interest in these state factors in a bigger way than it seems like there's been a lot of interest in them. And I was wondering if you'd thought about applying these to other areas or where your interests with these are going in the future.
Yeah, so that's a great question. I think the best way I can explain that from like a personal thought of where I'd be going with it is I realized working on this project and then my PhD or my postdoc work, which was trying to develop isotope systems for paleoclimatology, and using gradients to develop those systems, is that I'm really interested in understanding gradients. So I'm not, I guess this is a little bit of a cop-out, and I'm going to argue that pandemic brain has meant that I haven't thought much very deeply about some of this. I'm not sure where I want to take state factors and thinking about them personally, but I know that applying gradient understanding has really helped me think about not only the natural world, but also social issues. And so really trying to get students out looking at gradients where we have fairly consistent changes, where there are abrupt changes, what that means for the system. So is that a slightly evasive answer your question answer?
No, that's great. And as someone who works on southern gradients, that's also, really, really interesting to me. Any questions from the group? And just invite questions of all types from folks. Just drop them in the chat or feel free to unmute.
Not a question, but just a comment. I really liked the layout of your presentation and that you used a lot of drawings and diagrams. It was really helpful for my brain. So thank you for that.
Oh, thank you. I must admit, I must thank my field help / partner in crime, Harmony, for helping me with this. When organizing that, she had the idea of, especially for the little, to help people stay in line with my talks, is having a little cartoon that then pops up in the corner and stays there so that people are like, oh, if I got lost, I remember that cartoon kind of means this thing, I can follow again.
I don't have a question, but just also a comment. I really enjoyed my name is Joseph. I really enjoyed your presentation. And again, just like Bailey said, really nice layout. The other comment I have is that it was really nice to see somebody work on Hawaiian salts. I've been touting those salts as great chronosequences to my class for the past 10 years, fundamentals to salt science. So thanks for verifying that I'm on the right track there somehow.
Oh yeah. I love working on Hawaiian soils. I'm hoping I can keep doing it now that I'm on the East Coast again. But so coming up, my undergrad advisor did his PhD in Hawaii. So that's where I started thinking about soils and ecosystems. And one of the hard parts about it, though, is thinking like, okay, this is allophanic soil that's not actually a very widely distributed soil around the globe. So how do we making all these arguments, right? They're like, oh, like these are pedogenic thresholds, it's a broadly applicable concept. How do I actually get away with that? Excuse me. And luckily, Dr. Dixon, who I think she's at Minnesota now, she did her postdoc with Oliver, and I was actually able to help with this project, looking at New Zealand versus Hawaii. So using New Zealand's non-allophanic, more temperate soil, right? So don't actually remember the parent material, but I think it's like sedimentary aeolian deposits, and some igneous melange, but just being able to look at like, where do the thresholds still line up? Where do they not? Is this type of conceptual thinking actually applicable broadly? And by and large, it does. But the big difference is that because Hawaiian soils are so much volcanic glass, they weather so much faster than other minerals. So they're much more easily weathered is the difference.
Yeah, it's a great chrono sequence. I hate to be rude, but I have to rush into another meeting and another platform. So I enjoyed your talk. Thank you very much for giving us that talk.
Yeah, thank you.
Have a good one, and hopefully your cough is clearing up soon.
Yeah, thank you.
All right, I have one more question if no one else has a question. I guess I was talking with someone recently who's working on iron and aluminum organic interactions. And she was looking at soil types on a continental scale and seeing that andisols and spodosols can sometimes store a disproportionate amount of carbon in terms of land area. And I was wondering if you could hypothesize. I know you didn't show a lot about carbon, but yeah, where do you think the carbon's going to be hanging out in your system?
So in that, the carbon is much more stored in the sorry, I'm going to stop share if that's all right. It's much more stored in iron organic compound like interactions. And that's partially because that as you get wetter and you have organic matter developing, and so that there is more organic matter to store, the leaching of the aluminum means there's not as much aluminum around to have that interaction. Yeah.
Right.
Yeah.
But I guess you think it would behave similarly to some of the other variables you were looking at, where there's like a point in which carbon storage is going to be more significant, I guess maybe in some of the iron complexes, or is there a sweet spot there?
So actually, if you're willing to wait a second, I have that figure somewhere. Unfortunately, I can't think of it off the top of my head.
That's totally fine.
But yeah, so it is a changeover where you're moving from iron or from aluminum organic complexes to iron organic complexes, and it happens, I believe, right around zero water balance.
Cool. Yeah, it's neat to think about that going into broader modeling efforts of, especially for organic matter and carbon.
Yeah, and Oliver and Mark Kramer I think in 2016, it's been a while, they published a paper on the leeward side of Mauna Kea, looking at carbon storage and short range order iron complexes mainly, I believe.
Great. Well, thank you again for an awesome talk. And if everyone can either do the emoji clapping or unmute and clap, can applaud you, Jesse.
Thank you. Thank you all for having me. It was wonderful to sit down and actually think science communication more and less of like, okay, have to prepare for a class.
Sure thing. Yeah, it was great. So thanks. And thanks everyone for coming.